Thank you, Nikki, and all the organizers for inviting me to present my work. Um, so here at Blackman Lab, we are studying both aspects of floral maturation traits in a field experiments. Um, so previously, it was the motivation for this work comes from the previous work from Stacy Harmer's lab and Blackman lab together, where they published this uh, uh, paper on circadian regulation of sunflowers and how this floral orientation and then pollinator visitation is integrated into this environment, and then how this internal circadian clock coordinate the physiology to influence growth and reproduction. So most of this background was already covered by Nikki and Karine, who already set the stage. That made my work a little easy. Though I want to just emphasize again on this, that the solar tracking has been previously shown that it integrates both light dark cycles and how this clock and then hormones come together to mediate this east and the west movement of this uh, uh, solar tracking shoots. Here we can see that by the anthesis, it's already the tracking is slowing down, and then at anthesis, it stops facing east. These aspects of heliotropism and the aspects of head orientation was previously shown by Nikki, Hagop, and in the paper. And one of the outcome is that was that the east-facing heads attract more pollinator visits, and that was clearly shown here in the experiments at Davis, as well as in Ben Blackman's lab in Virginia. And then the east-facing heads warm up much quickly in the morning compared to the west-facing. And then when using the supplemental heat to the west-facing heads attracted more pollinator visits. And then later on, recently, this was more narrowed down to the few hours in the morning, where Nikki closely looked at this insect counts in the first 2 hours of the sunrise. And again, it was replicated that during early morning hours, that east facing has advantage over the west facing, and it's the temperature that regulates this. In parallel, Blackman lab had worked on this advantage that had shown that east facing plants have more advantage with siring more offsprings, and though it did not affect the yield and other parameters of this. So one more aspect of this one is that the floral development is more advanced in the east facing heads compared to the west facing heads. The 3 stages were also like what we look here was that one is the bud break stage, and then followed by pollen star stage, and then the stigma elongation or the stigma curling stage. Most of these parameters were advanced. Most of these traits were advanced in the east facing compared to the west facing. So there is a remarkable consistency in the timing of anthesis after the dawn, again, which suggests the role of clock, and then how and the temperature effect on the whole process. Here is an picture, time-lapse picture that we collected from our field at Davis, where we see that all of this process is happening like in a frame of few hours in the morning. So as soon as the bud breaks, we see that anthesis happening, and then the male phase with the release of the pollens, and the bees come and collect that pollens, and then followed by the staminate phase. We tried to replicate this results in wild Helianthus annuus and populations, and we were able to do the same thing, capture the events using a time-lapse pictures or images, and then what we found was that east-facing heads also attracted more pollinators in the wild populations as well, and the domestic and they were all the east-facing was developmentally advanced compared to the west-facing during early hours in the morning when we looked at the percent florets that had put at the pollen star stage. So this was the motivation to understand further how this pollen, the entire floret maturation is regulated by sensitivity to both the environment and how this clock integrates into this growth aspects. So previously we have seen that timing of floret maturation is very important for the pollinator visitations and as a consequence on yield. However, how this internal and external cues affect this style and anther development is a larger questions being addressed by both Harmer Lab and Blackman Lab, and with collaboration with other labs. So here our focus is on to identify the genetic loci that regulates most of these processes, and definitely identification of these genetic loci would help in breeding traits that can augment more pollinator visits, also reduce sensitivity to the environmental extremes, and improve some of these agronomic traits. So with this background, we had two approaches to map this traits. One to go with genome-wide association mapping with the SAM panel, which has a collection of elite cultivars, land races, oil, different kinds of oil and confectionery varieties. And then this is a 264 lines collection, and SNP resources are already available for this, which were kindly shared by the Sunflower Genome Consortium. And in 2019 at Davis, we set out this experiment. And then the other part is involving the wild cross progeny of the wild cross between Manitoba and Kansas populations. The previous technicians and postdocs had put a lot of effort in generating this F4 and F5 progeny that we are using this in the current field season. Now it's in progress at Davis. And I would like to highlight a few results from the 2019 work at Davis involving the SAM population and how this came out with the GWAS studies. So we had 264 lines in 3 replicates, and it amounts to around 800 buckets of plants. And then we had some check lines as HA412, and then there was also borders to buffer. This is how our field look like, and then we prefer to grow the sunflowers in buckets so that we will film them facing east first and then rotate 180-degree to score the west facing heads. It's a pretty much bit of humongous task to handle everything at one time. So we just staggered the planting, and then uh, so we have also very limited number of cameras that we can set up in a day. So around 60 to 70 plants were filmed each day. Uh, um, as we see here, so we captured pictures of every 5 minutes interval, and then for a period of 3 days, and then turn it to the west. Some of these traits how were scored as that it's a time of the day when there is a 50% bud break in those pseudohorals that are maturing at that in a particular day. And then it was all like we also uh, did the same for the pollen star and style maturity. One of the derived trait from this would be the orientation sensitivity comparing east versus west. Overall, we collected around 1,150 videos of 72 hours each, which would make us have a video length of about 82,000 hours. And this is really a lot of pictures to look at or the videos to watch. And then the I couldn't estimate the phenotyping man-hours yet, how much, but we are logging in all of these tasks because it involves multiple people, enthusiastic undergrads who are helping me to score this. I think by end of this phenotyping period, we'll come to know how many hours were really invested in this, getting the data. Some other additional traits that were relevant to pollinators, like corolla length, width, and colors were also measured in the field there. So besides this filament length and style, color, and other parameters of the heads. Briefly, with this 264 diverse SAM panel accessions and with the resources of 650,000 SNP polymorphism mapped to XRQ version 1, we did two models. One is linear mixed model, and then the other one was a Bayesian sparse linear mixed model. Both of these models included kinship and PCA to account for the population structure. And then we also had a multiple testing like correction threshold for both linear mixed model and Bayesian sparse linear mixed model. And that's the stringency line that we establish for any SNP to be significantly associated with a trait. And we set out to map this Corolla length. I could show the nice clear normal distribution of the frequency in the panel for the Corolla length on the left side. So in the upper part of the Manhattan plot here is about a linear mixed model, which is the horizontal blue line is the cutoff for the multiple testing correction, which is little stringent. And then when it comes to Bayesian sparse linear mixed model, the probability inclusion p-value is equivalent to almost the negative p-value. We consider that as 0.01 across the scale. And we could see some significant associations on chromosome 1, 7, 10, 12. And when we looked at these candidates, we went into see like 50 kb upstream and then 50 kb downstream as a window to account for any potential LD in the panel. So some of these candidate genes were like kinesin motor domain, and then one indicated as annotated as maternal effect embryo arrest, and we need to probe more into this uh, in this region to see what other genes are there. So coming to the corolla width, we could map two two chromosomes, that is 6 and 11, which is clearly a nice peak in the Bayesian models, and the genes indicated in this region were all MIC type, basic helic, loop helic, and IAA related protein families. So the third trait we did was the stigma color. We could see that the stigma color here varies from like little light yellow to some intermediate to purple color. So we counted how many of these individuals are there in the whole panel, and we see that yellow color dominated the panel, and then very less of purple color. Then we tried to map this as well, and it's the genes, candidate genes included some B-box zinc finger nucleases, zinc finger proteins, and then GMP dependent kinases. So we were a little surprised by the purple color in this in the cultivated panel, and one hypothesis would be that it has been integrated by integration from when introgression from the wild that might have been happened, along with other traits that were carried out, like other agronomic traits that were integrated on chromosome 10, because we see huge peak on chromosome 10 here. So again, coming to this mapping Floret traits, we go frame by frame to look at each video for each plant that we had genotype, and then we noted all the bud break, pollen star, style maturity, and orientations, and we'll derive this orientation sensitivity as well. So head orientation sensitivity, we can use this on a trait basis. For instance, I'm showing here the pollen star phase relative to the sunrise, that all the red dots here are the east facing, and then blue is the west facing time difference. And then the y-axis is the each accession that has been phenotyped so far. Also, on an overall developmental basis, we can look at how this all the three stages are varies in the east versus the west facing flowers. We are trying to develop some implement multivariate modeling for this, so that will help in the mapping these traits. And I would like to present some very hot or preliminary results from a couple of these traits so far that has been phenotyped. So we can see here, this is a bud break timing, east versus west. We see there is a distribution going from minus 200 means, that means 200 minutes before the sunrise, and then plus indicates that after the sunrise here in this x-axis. And mapping, I just mapped using a Bayesian sparse mixed model for this preliminary analysis, and the candidate gene here on the chromosome 1 is auxin responsive family protein. So pollen star timing was the next phenotype, and we could see very clear results here on a clear peaks in this Manhattan plot, one on chromosome like 1, and then 4, 5, 10, 11, and 13. So this indicates that there are several loci that could be controlling this trait. And the candidate gene in this window was related to GA signaling, and then there is some kinase domain protein which is involved in regulating the auxin signaling as well. So GAs, we are a little intrigued by the results of the GA signaling being associated with this. And in Arabidopsis orthologs of this work has shown that, for instance, mutation in GA biosynthesis and signaling would reduce the length of anther filaments as well as the style length. So the first is about GA oxidase, and then GA1, and then these are the GID1, the triple mutants with the it's a receptor knocking off the receptor. So it's not only the GA is relevant, has been shown in Arabidopsis, but also in the sunflower dwarf mutant, which was shown to be a GA deficient line. And here the investigators also noticed that there is a limited pollen viability in these dwarf lines, and then the pollens remain inside the flower because there was no anther elongation happening in these dwarf lines. However, the exact loci of this or the locus of this mutation is unknown yet. Another evidence implicating GA as a promising candidate here is comes from a work from Lobello and co-workers, where they show how this GA suppresses the filament elongation, and they compared auxin and then GA treatments here, where the filament length was decreased, were suppressed, elongation of the filaments were suppressed with the GA3 treatment. And they tested across various hormonal concentrations for each of these hormones. And then the upper panel here is about a constant white light for 24 hours. When it comes to the alternating light dark cycles of 16 hours light and 8 hours dark, there is more dramatic effect of GA treatment on the filament length, which would indicate that the which would indicate integration of this phytohormones, and light dark cues can potentially happen at this GA signaling levels. So we also map briefly this style maturity timing, and the candidates, the SNP associates were on chromosome 10 and 14. That includes one of the shaggy related kinases. And then I need to probe further into each of these window to see any other candidate genes here. So the other aspect that we are working here is to map the pollinator trait as more pollination related traits. What happens is we are filming the pollinator visitation as well, along with the floret maturation traits. They are we see a diverse bee community or visiting this sunflower lines that we grow in every season. And then we also plan to map this kind of this as a pollinator visitation as well as a trait in jivas, and that's our plan to integrate it. However, it's very laborious to count the pollinators in the 1,200 lines that we have, plus the wild panel that's coming up, which is equal in sizes with 800 plants each. So one idea was to use some kind of machine learning tools uh, uh, to count these pollinators in the images that we have. Sometimes the pollinators can camouflage with the background, and it's very hard to detect even with the visually for us. So I in with the help of artificial intelligence and machine learning experts, Nikita, we are planning to develop a tool that can count the pollinators in each picture frame that we have collected. This is a preliminary model, and it's in progress now. It's a only 100 images have been trained so far, and then using this algorithm, machine learning algorithm called as Yellow version 2, we were able to identify the bees with 100% confidence, though they are really hidden and camouflaged within these florets. You can see there are two examples where it's very hard for us to recognize, but the machine learning tool is very powerful to be able to recognize this. So this is at this point we are only able to classify like bee presence or absence. We would like to go further and try to map these diverse bee communities so that we get the data out that what are the proportion of these diverse visitors that are in the sunflower panel. So if we also seek some of these images from the community, if they know the species as well as like on the images of insects on sunflowers, we would like please contribute it to us so that we can train the model using those custom images and get the readout of this in the machine learning tools. With this, I would like to acknowledge my colleagues who are great support in organizing and for the success of the fieldworks in 2019 and this year as well. Ryan, Tris, and Anna, who are working closely on this project. And of course, the Floret scoring team of very enthusiastic undergraduate students who worked already almost like 2 years on scoring, and also students who helped me in setting up this field, filling the buckets and everything. And I would like to extend my thanks to Ben Blackman for giving me this opportunity to work on this project, and lab members Ali, Jack, Peter, and Lauren, who are there to help me whenever there is a need in the field work as well. And extend my thanks to Harmer Lab, Jessica Barb Lab, and Lara Marek from USDA. Thank you.